Welcome, everyone, and thank you for joining us today for our webinar titled From Mouse to Monkey, Revolutionizing Research via Preclinical Continuous Glucose Telemetry. This is Liam Samuel from Inside Scientific, and I will be your host for today's event. Our session is sponsored by Data Sciences International and will feature scientists presenting a new method for monitoring blood glucose via implantable telemetry. This technology allows a complete continuous glucose profile to be observed, reducing the need to take repeated blood samples, minimizing stress for the animal and labor for the researcher. First, we will hear from Dr. Stephanie Simmons, NHMRC Peter Doherty Biomedical Early Career Fellow and National Heart Foundation of Australia Postdoctoral Fellow. Her research aims to expand our understanding of obesity, diabetes, and cardiovascular diseases in order to expedite the development of diagnostic tools and pharmaceutical agents. She has been published in top-tier journals including Nature, Cell Metabolism, and the Journal of Clinical Investigation, and has been acknowledged for her innovative research with numerous prestigious awards. Following, we will hear from Dr. Megan Fine from the Surgical Services team at DSI, who will present the advanced surgical model that has been developed for the use of glucose telemetry in mice, rats, and larger animals. Advice for techniques that can help improve the surgical success rates and telemetry signal quality will be provided, and calibration best practices will be shared. Finally, she will discuss concurrent collection of physiologic data from multiple telemetry implants in the same animal.
Thank you. Thank you for the opportunity to present. So I'm an early career researcher at Monash University within the Monash Biomedicine Discovery Institute in Melbourne, Australia. And I'll be talking to you about the experimental application of the DSI continuous blood glucose monitoring system in mice. This system has become integral to our research as we're particularly focused on understanding how chronic diseases develop in obesity, and one of those diseases being the dysregulation of glucose tolerance. So we've used DSI equipment for over 9 years, and my partnership with DSI began when we were trying to understand how elevated blood pressure and heart rate developed in obesity. I began using the HD X11 probes, which measure blood pressure, heart rate, and ECG activity, as well as the PAC10 probes, which measure blood pressure and heart rate. And so these probes became integral to one of our publications showing that hormone produced from fat cells, being leptin, acts in the brain to elevate blood pressure in obesity. So when DSI developed the HD-XG, continuous blood glucose probes, this allowed us to then gain an understanding in a chronic setting where mice were non-stressed, living in their home cages, the chronic regulation of blood glucose in an obese animal. So we now use this technology readily, and we've used it combining with a number of physiological techniques. I should probably just talk about the surgical procedure is not remarkably different between the implantation of the blood pressure and the blood glucose probes. The flexibility of the catheter on the blood pressure probes is a little bit different, but it doesn't change your surgical approach dramatically. We also find no additional complications with recovery or surgery from either implantation. So as I said, we look at the disease state and particularly that being obesity and how that links to the chronic diseases of cardiovascular disease and diabetes. And so one of our key models within the lab is a diet-induced mouse model. The reason we use mice is because they readily become obese, but also because of the genetic manipulation we can produce to this type of rodent model. So to make mice obese, we put them on a high-fat diet for 20 weeks, after which we show that they have a significant increase in body weight, and this is mostly made up of increased body fat. So we've implanted the DSI system into obese mice and lean mice and found no complications in putting them into an obese animal compared to a lean animal. So we've used the continuous blood glucose system to measure kind of the phenotype activity of the system in lean and obese animals. And what's really great compared to just doing a manual blood glucose, say, phenotype analysis, is you get continuous recordings, and you get this resolution that you would not get if you were using a manual blood glucose assessment. So you can see you get a lot of time points during both the light and the dark period. You can understand the rhythm of blood glucose. You can compare this to food intake, and you can also remove food and watch what happens with the blood glucose levels. You can see here between the lean mice in blue and the diet-induced obese mice in black. The diet-induced obese mice have elevated blood glucose, but they also have a difference in rhythm, with both animals increasing in the dark period compared to the light period, but also differences in rhythm when they're increasing and decreasing. Additionally, we've done experiments where we have put animals on a chow diet, had the continuous blood glucose probes implanted, and then we've switched these animals to the high-fat diet. And one of the great things I like about the continuous blood glucose system from DSI is the ability to pick up subtle changes in blood glucose. And I think this is evident here, where you can see that you get a subtle increase in blood glucose. Nothing that would, when you took one data point, that you'd necessarily see. But when you have a large amount of data and accurate data, you can kind of get patterns and start to understand the resolution that's occurring. Additionally, the system allows you to measure core body temperature and locomotive activity. And this just allows you to have a much greater understanding of the whole physiological profile of the system. One of the other major benefits of the continuous blood glucose system is its utilization during a GTT. So we do manual glucose tolerance tests, and we might get 6 recordings or 6 measurements of blood glucose. With the continuous blood glucose system, you can get much more data. So you can see here, we have done an experiment where we have pre-treated mice with a drug at negative 60 minutes. While you can record changes between negative 60 minutes and 0 time, you don't get a description of the timing of that change. At time point 0, all groups received a bolus of glucose. And again, you can appreciate a change between blood glucose at 0 and 30 minutes. But again, the timing of this change is missing. If we do the same experiment, but utilize the continuous blood glucose system, you can see the degree of data and the information acquired is remarkably different. So example is the compound B in light blue compared to the compound A and B in dark blue. And you can see that, yes, they both decrease in blood glucose, but the degree with which they decrease is remarkably different, and the pattern of this decrease is remarkably different. Similarly, the rate of increase following a bolus of glucose in the measurement of blood glucose is completely different between all treatment groups. And I don't think you get that information when you do a manual analysis. And so you don't really understand what your drug is doing till you have this kind of information and this kind of rate of increase, rate of decrease, and put it together with the rest of the physiology that's going on with other hormones and other systems, that you can really understand truly what's happening in the animal. So the unparalleled temporal resolution of the DSI that the DSI probes provides us gives us a greater insight of how drugs work and the physiological systems affected. So another great use of the continuous blood glucose system we've found is the use in understanding how hormones affect the glycemic control of animals. Over 10 years ago, this paper was published in Diabetes describing how leptin given to rodents in the intraperitoneal cavity could reduce blood glucose. But the timing and the rhythm with which leptin caused this decrease could not be measured at the time, and it would be really hard to assess 20 years ago. But now with the DSI continuous blood glucose system, we can get a nice baseline recording, and then we can administer leptin in the same way, and we can understand the timing and the degree of decrease that leptin is actually producing. Additionally, we can understand the rest of the physiology that goes on to re-increase blood glucose and to counteract leptin's effects on blood glucose. So the versatility of the DSI continuous blood glucose system has allowed us to combine it with other innovative physiological techniques, including chemogenetics. So the ability to give a virus and change the activity of a certain neuron or neurons expressing certain key markers. So I should just point out the following studies are under review at the moment, and hence we have used X to describe the genes we are actually talking about. So we have taken X-cre mice and we have implanted the continuous blood glucose probes. Following baseline recordings, we've injected mice into a region of the brain called the Dorsomedial hypothalamus, or the DMH. And this is with this chemogenetic activator virus. Following a recovery period, the DSI equipment has been used to measure blood glucose, core temperature, and activity continuously. And what you can see is following the injection at the arrow here of a synthetic compound, IP, we can cause activation of the neurons in the brain that have been infected with the adeno-associated virus. And when we do this, we cause activation of just these neurons that express a certain receptor, we can get a decrease in peripheral blood glucose control. And the resolution by which we can show this in is quite remarkable. Additionally to this, we also get core body temperature and activity recorded, and so we can understand more of the physiology that's going on. We can combine the previous experiment with a glucose tolerance test. And so doing the same protocol as we did before, but while also injecting this period after the activation or the IP injection of either to cause activation of the neurons, we can give a bolus of glucose. And this gives us a challenge to the animals and lets us understand how the removal of excess glucose is occurring in, I think, remarkable resolution. And at the same time, we're measuring locomotive activity and we're measuring temperature, understanding more of the system than I think we have ever before. So one of the reasons we use mice is the ability to utilize the Cre-lock system. And this includes using mice that have flox receptor neurons with a stop codon, with which we can give an adeno-associated virus into specific brain regions, and we can knock out the receptor expression on particular neurons. So hence in these experiments, we have taken X lox mice and we've injected them with adeno-associated virus into, a Cre-expressing adeno-associated virus into the Dorsomedial hypothalamus region of the brain. On the left here, you can see what we have to do when we do a manual glucose assessment. We take a baseline glucose tolerance test. We take it again after 1 week and again after 3 weeks. And we do get a profile of what's happening with the animals, but they're under probably more additional stress, and we're also having to manipulate them or touch them a lot more than we do with the continuous blood compared to when we use the continuous blood glucose system. On the right here, you can see the same experiment when we use the continuous blood glucose system. Additionally, the amount of information we get doing the same experiment but using the continuous blood glucose system is remarkable. The other reason I like using the continuous blood glucose system is we can collect data continuously. So doing the same experiment, but just leaving the system on while the mice are on the plates and in their home cages, you can understand their daily blood glucose profiles. And we've found when we've knocked out the receptor expression on certain neurons in the brain, we can show that the basal blood glucose level slowly increases over an 18-day period. If we were to do this via manual analysis, we might have 6 time points per day. Here, after 18 days, you could end up with millions of time points, and it gives you the profile and the rhythm of what is actually going on. So I should explain this experiment a bit more, sorry. So in black, we've injected mice into the dorsomedial hypothalamus region with an AAV into X-expressing Cre receptor mice. And in purple or pink, we've given a scrambled AAV, so a control AAV into the same mice, into the same region to act as our control. And in both animals, we're measuring continuous blood glucose over the 18-day period. From that data previously, you can also take out the 24-hour profiles, and you can understand both the rhythm of blood glucose and how this might change, and you can understand, compared to from day 7 to day 14 to day 18, how the rhythm is changing within the same animal. So it gives you both the comparison within the same animal, but also with the control group. So thank you very much for listening to this presentation. I can't endorse this equipment highly enough. I think it really increases our ability to answer really important physiological questions without the stress, with increased accuracy, without the bias and variability that human manipulation to an experiment can provide. It also, in certain circumstances, eases the burden on the researcher in a way that you are recording continuously and you don't necessarily have to be present to do that. I thank you for your attention and look forward to answering any questions you might have. Thanks.
Today, I'll begin by explaining the value of using telemetry to monitor blood glucose. Then I'll provide an overview of the surgical techniques used in the mouse, rat, and large animal species. I'll share some of the lessons we've learned about calibrating the implants. And finally, I'll touch on some ways to combine additional cardiovascular endpoints with blood glucose telemetry, and then save some time at the end to answer questions. Continuous glucose telemetry allows for improved animal welfare, which is critically important to me as a veterinarian. I'll go into this topic in a little more detail during the next slide. Because researchers can now see second-to-second glucose data, they're able to identify trends and gain insights never before possible. This technology has led to the ability to improve study design and has allowed for cost efficiencies, especially through time savings when compared to more traditional experimental techniques. In our hands and in the hands of other researchers, we've seen some major refinements that include the ability to collect continuous data with minimal animal handling, which decreases animal stress and increases welfare. Reduced data variability helps provide greater statistical power, and this abundance of quality data leads to a reduction in the overall animal numbers used studying the diseases of glucose metabolism. There are some important principles that hold true for glucose telemetry surgery across all species. One of the main requirements for signal longevity is keeping the tip of the sensor where the blood glucose is actually being measured in freely flowing blood. We recommend inserting the sensor into an artery distally and making sure that the tip sits in a large central artery proximally, such as in the aorta. We found that it's helpful to hydrate the sensor prior to insertion. This will help decrease the amount of time it takes for the sensor to begin providing accurate physiologic data. We place the sterile saline on the sensor tip at the beginning of the surgery so that it's well hydrated by the time that we're ready to insert the sensor. Following surgery, we typically wait for about a week for the animals to fully recover before we perform the first calibration and place them on study. Because mice are the most frequently used research model, I'll start by talking about glucose surgery in the mouse. There are a few important preoperative steps that I'll describe to help ensure successful surgeries. Because we're going to be ligating the left carotid artery for this surgery, we're relying on the circle of Willis to provide blood flow to both sides of the brain. In order to ensure that the strain that you're working with has this continuous blood supply around the base of the brain, you can ligate the left carotid artery and then recover the animal from surgery. If the animal doesn't show any abnormal neurologic signs, it should be safe to proceed. Next, we need to understand how deeply to place the sensor to ensure that that tip sits in the freely flowing blood. The first step to do this is to place a ligature at the point where the left carotid artery branches, which is labeled as bifurcation in this image. Then we'll open the chest and dissect out the aortic arch so that we can measure the distance between our suture and the top of the arch. In this example, the distance is 10 millimeters. We want the tip of the sensor to extend slightly into the aortic arch. Therefore, in this example, we're going to insert a total of 12 millimeters of sensor length. In order to plan for all of our surgeries, we take measurements like this from some representative animals near the top and the bottom of our weight range. The appropriate insertion depths can then be estimated for the rest of the cohort. This process should be repeated whenever you change weight, sex, or strain of the animals, or if you experience an increase in signal failure, because this could be related to clot formation over the sensor tip due to an incorrect depth placement. There are two potential locations to place the implant body itself in the mouse. Intraperitoneal placement is only recommended for mice greater than about 23 grams to make sure that they're large enough to comfortably accommodate the implant. The benefit of IP placement is the ability to obtain a true core body temperature, which helps provide the most accurate calibration possible. This is, however, considered a major surgery. Subcutaneous placement can be successful in animals down to about 19 grams. It's a less invasive surgery, but temperature can be more easily affected by environmental influences. And if the pocket is not created large enough, you can see issues with skin irritation or necrosis. The first step of the surgery itself is to isolate the left carotid artery. We use the left carotid because the carotid and subclavian arteries branch off of the aorta separately on this side, whereas they originate together on the right side as the brachiocephalic trunk, and we want to avoid disturbing the blood flow to the forelimb. So for orientation, in this slide, I've flipped the animal 180 degrees, so now we're seeing it as the surgeon would while they're doing surgery. The animal's head is towards the surgeon and the tail is pointing away from the surgeon. Once the left carotid artery is isolated, it's ligated at that bifurcation, and 2 additional sutures are placed distally for hemostasis. Next, a 25 gauge needle is bent backwards to 90 degrees, and this will be later used as an introducer for the sensor. There's a white marker on the sensor approximately 12 millimeters back from the tip. And the surgeon is going to use this marker to determine how deeply in the vessel the sensor is placed. The exact placement of the marker should be measured before sensor insertion because accuracy down to even 0.5 millimeter is important in this surgery. We always recommend using a vessel cannulation forceps to hold the sensor to prevent damaging it. The sensor should be held in the channel of the forceps, sitting up on its edge, as you can see here, and not laying down flat. This is going to allow the sensor to bend the correct way as it goes into the aortic arch. The sensor should also always be grasped behind that exposed enzymatic well, grasped near where the arrow is shown here. With the sensor properly grasped in the vessel cannulation forceps, the artery is punctured with the bent needle, and the sensor is passed under the needle tip to the predetermined depth. The white marker on the sensor is used to determine how much sensor length has been inserted. In this example, we're going to insert until that white marker at 12 millimeters lines up with the suture at the bifurcation. Once the appropriate depth is reached, the caudal and middle sutures are tied tightly around the vessel containing the sensor, and the tails from the cranial ligation suture are tied around the sensor just before it enters the artery. It's important to make sure that the sensor lies flat underneath the skin of the neck to prevent any irritation. With intraperitoneal placement, the device body would have been placed inside the abdomen first during a previous step, and then the sensor would have been exteriorized and routed to the incision over the neck. For subcutaneous placement, a pocket is now made on the flank of the animal just in front of the hind limb after the sensor has been inserted and secured. It's important to keep the pocket very lateral with a subcutaneous placement so the implant doesn't migrate ventrally and drag on the ground, which could irritate the animal's skin. The pocket should be quite large and very easy to accommodate a 3 cc syringe. Saline should be instilled into the pocket and then the device is slid into place. Next, we'll talk about rat glucose surgery. You can see that the implant is a bit different with a suture rib, which will be secured to the lumbar muscles, and an external reference electrode that's secured to the ventral body wall. For the rat surgery, we recommend placing the implant intraperineally and inserting the sensor directly into the abdominal aorta. First, a midline laparotomy is performed. Then the intestines are retracted and the tissue overlying the abdominal aorta is bluntly dissected away. The aorta is carefully separated from the nearby vena cava in 2 locations. One is just caudal to where the left renal vein crosses over the aorta proximally, and the other location is just cranial to where the aorta branches into the iliac arteries distally. After the vessels are separated, 2 pieces of suture are placed underneath the aorta for later occlusion. But no tension is placed on the sutures yet because we want to make sure that we minimize the amount of time that blood flow is compromised to the hind limbs. Once everything else is ready, tension is applied to both sutures and the sensor is inserted using the same technique as in the mouse. As the sensor reaches the cranial occlusion suture, a micropipette tip is used to dispense a minimal amount of vet bond needed to seal the vessel around the sensor and prevent bleeding. It's helpful to begin by applying a small drop of glue to the vessel directly underneath the sensor first, and then finish with small drops on the side and top last. It's imperative to avoid placing too much glue because excess glue can prevent adequate blood flow and cause hind limb ischemia. Once all the bleeding is stopped, 2 fiber patches are placed, and this will help encourage fibrous tissue growth, which is what holds the sensor in place chronically. The first fiber patch is placed on top of the sensor and secured with a small amount of tissue glue on all 4 corners and right where the material contacts the sensor. The second patch is placed underneath the sensor, but on top of the aorta and secured similarly. After the sensor is secured in the aorta, the connector will be sutured to the lumbar muscles next to the vessels to help prevent postoperative migration of the sensor. First, 2 sutures can be pre-placed into the muscle. And then the sensor is guided laterally, so the connector sits over the pre-placed sutures. The tails of those sutures will be drawn up through the suture rib holes and tied securely. Finally, the suture aid shown here can be used to secure the reference electrode to the inside of the ventral body wall prior to abdominal closure. Next, we'll talk about our newest glucose products that were just recently launched for large animals. They allow researchers to obtain continuous glucose measurements in freely moving, socially housed large animals. A variety of approaches may be used for implant body placement in large animals. An intramuscular approach shown in the dog on the left is popular and can be used in a variety of species. It's a good compromise between intraperitoneal placement, which is more invasive, and direct subcutaneous placement, which is more influenced by environmental temperature and can be more likely to cause skin irritation. A peripheral artery, such as the femoral artery, is typically selected for its large size and ease of access. The vessel is usually ligated distally, or a purse string suture may be placed if the artery is large enough to accommodate it. Similar steps are taken to insert the sensor as I discussed in small animals, but here a 20 to 18 gauge needle can be used to create a larger opening in the artery. The sensor is inserted so the tip lies in a larger upstream artery, such as the abdominal aorta. The large animal sensor does not have a suture aid on it like the rat implant does, which allows a greater length of sensor to be inserted to ensure that the sensor tip can reach the freely flowing blood in these larger species. Once the sensor is inserted to the appropriate depth, it's important to fully secure it so it doesn't migrate postoperatively, which could lead to clotting and premature loss of the signal. I've shown a series of friction knots here, which is what I use to secure it, including a Miller's knot first and then a finger trap pattern. And these sutures are tied around the sensor using the tails from the ligation suture. In addition, the 2 proximal sutures are also secured tightly around the artery containing the sensor. Now I'll provide some suggestions for successful calibration that we learned while we were developing and using the glucose telemetry in our lab. Calibration is needed to transform the data from the nanoamp output of the sensor into a more biologically meaningful value of blood glucose in milligrams per deciliter or millimolar. A multi-point calibration is performed by first taking a fasted baseline blood glucose reading using a handheld glucometer, then administering a bolus of dextrose, and taking a second glucometer reading near the peak. The timing of the second sample is determined by watching a live telemetry signal throughout the procedure. And for more specific details, we do have a technical note on our glucose telemetry website. We typically perform the first multi-point calibration about 1 week after surgery to allow the animals to fully recover and the signal to stabilize. You can appreciate in this data the return to a normal diurnal rhythm on this image. In addition, a multi-point calibration is also typically performed near the end of the study, and single-point calibrations are recommended approximately once a week to ensure continued accuracy. When working with mice in our lab, we've had the greatest accuracy for calibration when using saphenous vein samples. We found that samples obtained by tail tip amputation failed to accurately reflect the true degree of blood glucose excursion that we were able to appreciate using the telemetry sensor. The technique shown here allows for ample volume to perform blood glucose measurements in duplicate. In rats, however, we were able to successfully use either a small gauge lancet or hypodermic needle to puncture the very tip of the tail for blood sampling. This was made possible because the telemetry technology allows us to minimize the required number of blood samples. The technique is well tolerated by the rats and avoids the need to anesthetize them to amputate the tail tip. It may not be appropriate, however, if a larger number of samples were required. In the large animal species, the calibration procedures have been performed both under general anesthesia and using vascular access ports, which can be implanted at the time of the telemetry surgery. The use of glucose telemetry technology has opened exciting doors for researchers. Scientists are now able to combine highly sophisticated metabolic and cardiovascular endpoints in one animal. This image depicts a dual implanted rat with an HDS11 implant monitoring systemic blood pressure and electrocardiogram, and an HDXG implant monitoring blood glucose. Both devices also monitor body temperature and activity. Our newest product, the M1G, monitors these same cardiovascular and metabolic endpoints using one implantable device in large animals, providing both refinements and reductions and contributing to animal welfare. And I'd like to thank everybody for their time today. I look forward to hearing your questions in the Q&A session to follow.
Again, thank you both for sharing your research today. Now I'm going to ask Dr. Simmons and Dr. Fine to join the audio line, and we will transition into a Q&A session. We've got some great questions up today. So the first question is, how long does the sensor last? Megan, can you answer this?
Sure. So the device is guaranteed for up to 28 days following implantation. However, some devices will function for longer than that, up to 6 or 8 weeks. It's because the sensor works through an enzymatic reaction. So that gets used up as it's in contact with blood glucose. So if it's the animals at a really high blood glucose or extremely hyperglycemic, you may get more towards a 28-day sensor life. However, if it's a euglycemic or maybe a more mildly hyperglycemic animal, you might get a longer duration. So one tip that I can provide is that it's really important to keep the implant turned on after surgery. So unlike some of our other telemetry, say for example, a blood pressure device where you can turn that implant off to save battery life, the limiting factor with the glucose is more related to the sensor life itself versus the battery life specifically. So you don't actually want to turn the battery off. You want to keep that on so the device functions and the metabolites from the enzymatic reaction keep getting moved away on the device while it's on. So ideally you just implant it and then leave it on during that healing period until you start the calibration process, and then start the animal study after that.
Great. And Stephanie, what is your personal experience?
Yeah, so we've had no problem with probes not lasting in the models we use, being mice, lean and obese. And yeah, like with the blood pressure system, you often can get a couple more weeks than DSI promise kind of thing out of the probe. So it's pretty good value for money, I feel.
Perfect. And the next question is, Dr. Simmons, in your experience, are there any specific difficulties in the implantation of blood glucose probes in lean versus obese mice?
No, we often get asked because of the excess fat in the obese animals, is it harder to get the probes in and things like that. The probes sit really well. The animals don't find any really adverse effects being obese and having the probe there. Probably the hardest thing is just locating the carotid artery and getting it in cleanly in that area, and very similar to what you'd experience when you're doing the blood pressure system as well, though.
That's great. And the next question is, Dr. Fine, can you please explain or clarify again how to do the calibration?
Sure. So for the calibration, it's essentially a glucose tolerance test that you're going to use typically. And we do, I think I mentioned in the webinar, but we do have some great resources on the website as well that go into even more detail than I will. But basically, we typically use a fasted animal for a few hours, so they're at a steady state baseline glucose. Then we use a glucometer to take a baseline value of the blood glucose. We actually sell a preclinical glucometer, so it's made by Nova Biomedical, called the StatStrip Express. And it's a glucometer that has an extended range up to 900 milligrams per deciliter. So if you're using, say, for example, a really diabetic animal, some of the other monitors will just like peg at a high reading and you don't actually know what your true blood glucose is. So this one will actually give you a true value, and it corrects for hematocrit, which is nice because other systems just assume a constant hematocrit for probably not a mouse species, so for another type of like a human species. So it provides the accurate results. But anyway, so you take a baseline reading and then administer the dextrose. And typically in mice and rats, we'll typically do an intraperitoneal injection of glucose, of dextrose. And so then you wait and watch the signal following the telemetry signal live. And then once you see that blood glucose value on the telemetry system peak, you wait about 3 to 5 minutes if you're doing an intraperitoneal tolerance test or oral tolerance test. It's a little bit longer of a delay for an intravenous tolerance test, about 10 to 15 minutes after the peak, because of the different patterns of absorption of the dextrose. And then you take a second sample near peak, and then you use those 2 samples to outline your curve and perform the calibration of the implant. Then that data can be input into the computer, and that allows you to transform into your milligram per deciliter or millimolar blood glucose values.
Perfect. Thanks for sharing that, Megan. And then the next question is, what animal models have you implanted with glucose telemetry? And as an extension, are any of the diabetic models more difficult to work with? Megan, maybe you could share your experience first.
Sure, I can start and talk a little bit about what we've done in our lab. So in mice, we've done just regular Swiss Websters, C57 blacks, and then we've done some metabolic models such as the OB-OB, DB-DB, and the diet-induced obesity, which was mentioned earlier. So both the animals control and then as well as animals that were on the high-fat diet. In rats, we've used Sprague Dollies. We've also used Sprague Dollies as a type 1 diabetic model that have been treated with STZ. And we've done fatty Zuckers, ZDSDs, and then in large animal, non-human primates, swine, mini pigs. So as far as if the diabetic models are any different or difficult to work with, that was one thing that we were concerned about when we started working with them, just because knowing that in general, diabetes tends to negatively affect healing. And so there are a couple things that we've done to help kind of mitigate those effects, and a few things that we found. So for one, if it's like a type 1 diabetic model, so something that we would induce like with a STZ induction, we typically try to wait till after the surgery to induce the disease state rather than inducing first and then doing the surgery on a disease model. Where that's not possible necessarily, like in a type 2 model, we just really watch them carefully post-operatively for wound healing, skin irritation, just general health and animal welfare. We have seen some animals that have issues with skin irritation or erosion, either over the sensor itself. That's why I mentioned that the sensor in the neck area. So you need to just make sure when you're doing the surgery that it lies down nice and flat underneath the skin. If it bulges up and you haven't laid it down nicely, you can get some erosion over the sensor there. And then when making the pocket so if we're doing a subcutaneous device placement, we want to make that pocket really big. In a mouse, it's pretty hard to make the pocket too big, but it's relatively easy to make it too small. So we just make a really large pocket on the side of the mouse, being really careful not to let that pocket open up ventrally so the implant doesn't slide down underneath the belly. We found in the obese models, there's a little bit of, there's fat kind of everywhere. And so it does provide a little bit of padding under the skin. So we've actually seen typically less skin issues with those obese models than we would with like a C57 black that can have a little bit of skin sensitivity anyway. So sometimes in the leaner models, we end up putting the implant intraperitoneally to help protect the skin. It also, like I mentioned, gives us great core body temperature, so we have really accurate calibrations as well. But just a kind of a difference between the lean and the obese models.
Great, thanks. And Stephanie, what animal models have you worked with?
So we've only used the mice models, all on a C56 background, lean and obese. We haven't gone to the DV-DBs or OB-OBs yet. The only tip I would give from what Megan was just saying is I think putting the pocket as far on the side as you can is a great tip, especially in the obese models. Otherwise, because they have so much excess weight around the middle, it can drag maybe on the ground kind of thing. So putting it on the side is a really important part of the surgery.
Great, thanks. And so here's a question. What's the maximum glucose level that this implantable device can detect? Megan, can you answer this question, please?
Sure. So with the ability to calibrate it up to 900, we have been able to find that the device has like a nice linear curve up to 900 milligrams per deciliter. Sorry, I don't know that in millimolar off the top of my head, but so yeah, it can go quite hyperglycemic and should still be accurate up in that range.
That's great. Thanks, Megan. Here's an interesting question. If you're implanting both blood pressure and glucose in large animals, where do you suggest placing the catheter and sensor?
Yeah, great question. So with the new M1G, that's a possibility with one device. So we've done some of that work initially in that product. And so one placement that we were pretty happy with was using essentially both femoral arteries. So we placed the implant itself in an intramuscular pocket, specifically underneath the external abdominal oblique and above the internal abdominal oblique muscles, and then tunneled one of the sensor to the one femoral and the catheter to the other femoral. And when we inserted those, we made sure that we inserted them to different depths. So essentially the sensor was higher up in the abdominal aorta and then the catheter was further down, just we didn't want those two to end right at the same level. And so we controlled that by how much length we inserted, which we could tell by how much length we had out. So that was one way that we were pretty happy about it. Another option would be to use a different peripheral vessel. So say, for example, you could use one femoral and the carotid artery, if that's a placement that you're more comfortable with. I guess it's similar in the small animals. So not using one implant, but I showed a picture in one of my slides of a rat implanted with an HD-S11 to monitor blood pressure, ECG, temperature and activity, as well as an HD-X11 to monitor the blood glucose. And those are available on 2 separate frequencies. So those you can actually collect the data concurrently in one animal with those. And so for that implantation, we put the glucose implant inside the abdomen and inserted the sensor directly into the abdominal aorta. And we placed the blood pressure catheter into the femoral artery, so into the peripheral artery. And so when we had the blood pressure catheter placed first, and then we went into the abdomen to place the glucose sensor, we could actually see right where that catheter ended. And so then we just inserted the blood glucose sensor just proximal to where the catheter ended. So we kind of had them staggered in the abdominal aorta, and that worked well.
That's a great answer. Thanks, Megan. Now, Dr. Simmons, how many samples are required or recommended during a glucose challenge?
So I think we do it a number of ways. So we will do a typical glucose tolerance test before we start really collecting data, just to calibrate the system, as DSI, I think, recommend. And we will also, and so we'd at least take 6 points during that. Throughout whatever experiment we're doing, we take at least one data point a week with the manual glucose, and to calibrate or to check the calibration. But often it'll be a couple of points during the week. And then towards the end of the experiment, we will always do at least one more glucose tolerance test manually while the continuous glucose system is running, just to check if there's been any shifting in the system, which we don't find. If you're within that warranty period, we don't find a whole lot of shifting in the lean and obese models we use, but they're not at the higher end, as Megan was saying.
All right, thanks, Stephanie. Now this question came in before the webinar, but just to clarify, do the animals experience any problems when housed in pairs, such as tangling or any other strain on the wires?
Yeah, so it's a good question. So these are totally fully implantable devices, so nothing is external in the animals. So there's nothing to get tangled or strained on. The animals may be individually housed for data collection with the small animal implants, you'll only be able to collect data from one glucose implant at a time. So some facilities still require, or some research still dictates, that the animals would be pair housed. So in that instance, you could either do a non-implanted companion animal, so they have a cage mate that they're with that's not implanted, or you could implant two animals and monitor from one at a time. With the large animal solution, like I mentioned, they are able to be kind of freely roaming and socially housed. So those animals can be housed together if that's acceptable to the research. And yeah, nothing externalized, no wires or anything to get hung up with.
Great, thanks. Now, in the interest of time, I'm going to make this the last question. Dr. Simmons, what is the success rate of implantation for you in mice? Have you experienced any unique challenges with this?
No, so the use of the continuous blood glucose system, and I was a little bit, I guess when you do the first one, it's a little bit different than the blood pressure system, just the end of the probe and I think the flexibility of the probe, but we have very little difference between our blood pressure and our continuous blood glucose system in terms of the success of the surgeries and everything, and it's very high. So the one worry I often get from researchers is the continuous blood glucose system can't be reimplanted into mice as the blood pressure system can, but I don't think you need to really worry about it, because once you have your surgical technique down, you shouldn't be experiencing a need to reimplant into other animals through the battery life.
Perfect.
Yeah,
thanks.
so we expect about an 80 to 95% success rate. So depending on, like we talked about, some of the animals are more or less healthy depending on how diabetic they are. But typically we expect at the end of that 28-day period that you still should have at least 80% of the animals around and healthy, quote unquote, to be on study, and the sensors working. So we do recommend taking that into account and overpopulating if you need to, if you have, if assuming that you should be should be at about 80% or greater at the end of the 28-day period.
Perfect. Thanks so much to you both.